My job here today, I think, is mostly to give you a little bit of an introduction and a platform to Annaliese. There are a couple of things that I mostly focus on in my research here. The primary focus at the moment is around detrimental effects, behavioural and metabolic, of excess sugar consumption. And we do have both small animal and human work focusing on that at the moment at the university. But we're particularly looking at withdrawal effects at the moment. So we're making the assumption that in 5 to 10 years everybody will listen to public health and they'll start removing sugar from their diets. And what we're trying to do is to track what benefits, if any, we can actually see with that. But the other core area that we're working on is looking at how our habitual diets, what you eat every day or so, might influence your energy expenditure, or some people might refer to it as fuel utilisation. Really, in its simplest form, it's how we will release and burn the stored energy that we have. So most people are aware that we've got stored fats and stored proteins and stored carbohydrates. Our proteins are our organs and our tissues, so we don't really want to be breaking those down. So we want to understand how we might be able to best manipulate accessing our fat versus carbohydrate stores during exercise. And so what I want to do for at least 10 minutes is I'm going to give you a little bit of a breakdown as to how we do that and how we understand that we're doing that. So a little bit of the mechanism side of things and why we would be interested in it. And I'm going to hand over to Annaliese, who spent the last year for her master's thesis doing a bit of a literature search, doing some data collection, and who is still doing data collection up until around about February or March of next year. So look, I don't want to scare too many people. Don't worry, I'm not going to go through this too much. This is for any of the biochemists who might have come out on the weekend to have a look and listen to low carb. Because the biochemists, if you talk to them about fuel utilisation, this is what they're thinking. A very complicated mix of ketone bodies, alcohols, sugars, fats, how we utilise them to generate our energy, which is an ATP molecule. But really, when we come from a physiology or an applied biochemistry perspective, all we're really focusing on is these components. Because these are the ones that we can readily measure without having to jab you with various needles to take biopsies from tissues and to take bloods out and what have you. Effectively, what we're focusing on is the oxygen content, which is what you're breathing in, and the carbon dioxide, which is what you're producing during your exercise. And over many, many years of physiology research, the ratios with which these might be fluctuating, either at rest with you sitting here now or during exercise, have been back calculated to try and tell us whether or not you're burning fats or whether or not you're burning carbohydrates. And that's what we're particularly interested in at the moment. So how do we do that? A little bit of energy expenditure, or indirect calorimetry 101, as they say. Air temperature, for those of you who weren't aware, is roughly 78% nitrogen, which is an inert gas, which means we don't metabolize it. And then there's roughly 21% of the air we breathe is oxygen, and then a very small amount is carbon dioxide. And that's pretty standard. It might fluctuate a little bit if you go to altitude or go deep below seas, but for most of us at sea level, that's what we're looking at. So we then can attach you to a machine which measures not only the air temperature, but the air that's coming out of your mouth. And we can compare then the ratio of nitrogen, oxygen, carbon dioxide, and what you're breathing out to what we expect should be there in the air, and we can identify how much oxygen has disappeared and how much carbon dioxide is produced. So if you're sitting here not doing too much metabolic work, then there'll be only a little bit of oxygen being consumed, a little bit of carbon dioxide being produced. When we get you to do some cycling or some walking or some running, then there'll be more oxygen disappearing and more carbon dioxide being produced. And that's simply what we're measuring. This isn't a photo from our lab, but it does very much look like our lab. One of the things I'm realising as I develop as a researcher and I go to more conferences, I'm supposed to be constantly taking photos of people in the lab and the experiments that we do. So at the moment, I'm just stealing images from Google. But that is very much the setup that we'd have people come in, and what we've been doing for the last couple of years now. Previously, we've been focusing on genetic limitations to fuel utilisation, but that really explains very minute variances between our populations, and so we're moving around to more lifestyle things. So how do we know it? Most of you maybe might have heard people talk about your RQ or your RER. It's simply a ratio of CO2, carbon dioxide, to oxygen. And this is a ratio that people say, if you're at 1, you're mostly burning carbohydrates. If it's at 0.7, you're mostly burning fats, and somewhere in between it is a mix of fats and carbohydrates. It's a very simplistic approach to take, which we don't necessarily just rely on. We use some more first principle calculations that other people have shown us, and just for interest's sake, I've put them up there. But what we actually measure is we use those numbers to tell us the absolute rate of fat that you're burning and the absolute rate of carbohydrate that you're burning in grams per minute. And we can match that then to the workload that you're doing. So whether it be heart rate or power output or a cycling rate, we can identify that, you know, if you're working at this particular intensity, this is how much fat and carbohydrate you're burning. So I'll show you that as a picture, which everybody much further prefers. This is kind of what the data looks like when you get an individual into the lab. And this is real data. This is an individual. It's a student that we tested a couple of years ago to show that it's so simple. We teach this as a prac class to our undergraduates. That's how nice and easy the test is to do, and the data can be cleaned. The blue graph shows us roughly how much carbohydrates you're burning in grams per minute as we increase intensity. So down the bottom on that horizontal path, or what we call the x-axis here, that's how hard somebody is working as a percentage of their maximum capacity. And then on the y-axis here is the increasing amounts of fuel. So the blue graph shows at rest, so most of you sitting here perhaps would be at best burning about 0.5 gram per minute of carbohydrate and about 0.5 gram per minute of fat. And then if you do more and more work, then they'll increase depending upon how well you partition your fuels. So a few things we'd like to point out to individuals is that there's this concept called fat max. That's the maximum amount of fat that you can burn. And something that people for a long time ignored was the fact that after a certain intensity it can start to decline. Which means somebody working at almost their maximal capacity could be burning less fat than somebody sitting down doing nothing. Which is a concept which a lot of people took a long time to grasp and accept. Whereas if you have a look at the carbohydrates, the reason for that is because as you do harder and harder work, you shift your reliance more towards carbohydrates. So you're breaking down sugars and glycogen content. So what we're interested in though is this fat max level, but also at what level of intensity occurs. Because the reason is that a lot of people have shown it's quite dynamic. So you can shift it. We can make various adaptations to our systems or our lifestyle to make that number there increase, or can occur at an earlier intensity or at a higher intensity, depending upon various manipulations. So a couple of examples of some variations. This is just one particular study, and I am cherry picking at the moment, but because it gave me one figure that showed me most of the concepts I wanted to get across. This is probably where most of exercise science's knowledge was limited for quite a long time. And that is, what you can see here is in these top plots here, this is just people's fat oxidation rates as intensity increased. This one is whether or not you've been doing a lot of training. So the white bar is someone who doesn't do much training, the black one is someone who does lots. And as you can see there, it hasn't really varied much. This is the difference between whether or not someone's on a bike or if they're on a treadmill doing walking, running exercise. This is whether or not you're ingesting carbohydrates during your exercise. So as you can see, the black bar is if you're not ingesting carbohydrates, but if you are ingesting carbohydrates via a sugar drink here, then you pretty much inhibit how much fat you can burn during your exercise program. And then this one here is a comparison between males and females, showing that females have a greater capacity to access their fat stores at a higher intensity as well. Now, a couple of things that we like to note here is that the range is really small. So if you have a look at this and you see, you're talking about a change of 0.5 to 0.6. At best here is 0.4 to 0.5. For the large aspect, most people have accepted that how much fat you burn during exercise has nothing to do with weight loss or weight regulation. But we think that that might be because we've been ignoring one really important factor. And this really came to light a couple of years ago when Steve came out the first time and really inspired myself to go and have a look a little bit more closer at the habitual diet data. And so I just want to, I know Steve's going to probably touch on this as well, but I just want to give you some more selective data to show what I mean. In 2005, this paper here is quite often cited and referred to by most professors of exercise physiology around the world as one of the most comprehensive analysis of fat utilisation in the world that we have. 300 individuals, healthy volunteers in the UK, and measured fat mass under a range of conditions. They corrected them for sex, for training status, but they didn't do anything on habitual diet. But what I want to show you here, on average, from the 300 people, they were burning at most half a gram of fat per minute. So 1 hour of exercise, you burn 30 grams of fat. That's not going to impact your status at all, right? But the range here is somewhere, some people were only getting up to 0.2, some people were getting up to 1, and I was varying about half their intensity. So the easiest way to measure intensity if you don't have a heart rate monitor or anything is just 50% is when you go, you know what, it's somewhat hard. Right? It's not really hard, somewhat hard, about half intensity. Then this year, 1,100 athletes who have gone through the Gatorade Sports Science Institute over the last few years, both the USA-based Florida one and the UK-based one, 0.6 grams. Now these are people who are highly trained athletes. They will be international representatives in soccer, tennis, basketball. They have a whole range of different sports. They even have some NFL players in there. So that's 0.6. But then Jeff Olek was doing this faster study, which Steve told us about a couple of years ago, and showed us some what was then preliminary data that is now published. And in that, he's got some very well-trained triathletes who are carb eaters, and their average fat max is 0.7 with a range of 0.4 to 1. But the low-carb triathletes who are matched for training status, their average is 1.6. And their range is between 1.2 and 1.8, which means the lowest capacity to access fat in a low-carb individual was greater than the highest trained individual in the carb group. So what that suggests to us is that perhaps one of the biggest limitations that we've got to understanding the role of fat utilisation during exercise is that nobody has really cared to monitor habitual diet. And even in this paper that came out in 2016 from the Gatorade Sports Science Institute, that's a paragraph in the discussion. Of course, we haven't cared to worry about habitual diets, nobody has, and that could be playing a factor here, which no doubt was probably included in the review process after somebody talked about Jeff Olek's work. Right? So this is what we're looking at. And what we're particularly interested in is whether or not we can see this in a general population. Because that's another limitation that we've got. A lot of the studies will use really well-trained individuals, triathletes, athletes. These were healthy volunteers, but they were still pretty fit individuals that came through. And if we can untap that access to your fuel stores by manipulating diet, then maybe we can have a lot more strength towards the arguments of why regular activity could be influencing it. So I hope I didn't overwhelm you too much with the background information there, but I'm going to call Annaliese now to give you a rundown of what she's found in the literature, and then also give you a quick overview of some of the data sets that she's been collecting from the individuals who have been coming in.
Hi, so I'm Annaliese. I'm studying my Masters of Applied Science by Research. And when myself and Kieron first started chatting about potential projects we could be doing, we were going to look at fuel utilisation in general, and that's when we started discovering that habitual diet wasn't looked at very much, and if it was done quite poorly. So for the study we're doing, we got a grant from the Collaborative Research Network for Advancing Exercise and Sport Science, which is organised through Bond University. And we also got a donation from Low Carb Down Under. So this made the research that we're doing possible. So thank you. So Kieron's covered some of these. These are the factors that will affect fuel utilisation. So at the top we have habitual diet, which is what we're looking at. And then going in a clockwise direction, essentially it's probably the more studied ones. So training status and your ability to exercise, or how much you exercise, are the ones which have been most researched, because essentially the research in this began looking at athletes. So they wanted to see how they could improve performance as opposed to looking at general health outcomes. And then they moved on to looking at eating before exercise, whether or not you ate, the composition of what you were eating. And then there's also some work in genetics, but that's recently emerging at the moment. And then sex, age, and body composition hasn't had much research, as they generally don't tend to test females, and there's just not very much hard evidence for these. So we've conducted a literature review. So we looked at a whole lot of papers. We got 37,000 results, and we narrowed it down to 9. The criteria for these papers was that it had to be a healthy population, so people who were weight stable with no disease. Some high-fat diet studies look at diets with about 30 or 40% fat, but we wanted them above 50% fat and not eating too many or too few calories. Greater than 1 week, just so it was actually representative of habitual. And then looking at whether the pre-exercise meals that they ate before exercise had the same macronutrient ratios. So essentially they were eating a high-fat meal before exercising, as a lot of studies will put them on a high-fat diet for a week and then load them up with carbs right before they exercise. And then they weren't able to be fed during testing, and they had to look at the actual fuels being burnt as opposed to just performance factors. So from these, we ended up with 9 different papers. And even though we're trying to make these papers succinct so we can try to draw some conclusions, the papers are still quite different because there's so many different factors affecting fuel utilization. So the first main difference is length of dietary exposure. So the shortest one was O'Keefe, which was 1 week, and most of them were about 4 weeks long, which might show some adaptations, but there could be adaptations occurring after that point. You might notice that the Volek paper, I haven't put in this graph, it's in the next one, because it's so much longer. They had a minimum of a 9-month diet. So this is a clear outlier, which might explain why Volek did get quite a few different results as opposed to the other studies. The other main difference between the Volek paper is that they conducted their exercise test on a treadmill, which everyone else conducted them on bikes. And treadmills will make people have a higher VO2 max because they're using more muscle mass, so it increases their fat oxidation. The next main difference between them was diet composition. When you say a high-fat diet, it's not that specific. So on the x-axis, down the bottom, we have the percentage of fat, and then on the y-axis, vertically, we have the carbohydrate composition of the diet. So we know to be ketotic, you need low carbs as well as quite a bit of fat. So the red data points are the papers which people were in ketosis, and the blue, I put my colours incorrectly in that diagram. The blue are ones who aren't ketotic, but some of them also didn't measure them, which might explain some more results. So then also dividing it by the time, the length of dietary exposure, whether they were fed or fasted, and whether they were trained or untrained, you'll see that there's not many studies which are very similar, which explains why you get so many different results. So you'll have some nutritionists and some sports science saying that they're detrimental, a high-fat diet is detrimental to fat oxidation. Some will say it won't make a difference, and some will say it is wonderful at increasing fat oxidation. So from these papers, and in the groups which have 2, you'll see that the NK stands for not ketotic, so even within the same group, you have some on a ketotic diet and some which are not ketotic. So the ones circled are the ones which showed some difference in the results. These results aren't necessarily a huge difference, but there was a little bit of a change in the ARIA, which Kieron explained before, or the fat oxidation during exercise. But the way they measured this was different because there's multiple different exercise tests that people can do. In the Pogliagli study, had the highest carbohydrate composition, and the other 2 isn't not really clear how they were completely different to the other studies. So therefore, what are the results of the current literature that's out there? There are changes which have been detected in fat oxidation during exercise, but it's very specific and it's only at certain time points within the exercise testing. In terms of performance benefits, so whether someone will be able to run longer or faster, it's quite unclear. Some will say they increase, some will say they decrease. One of the other main problems which affects this is that a lot of people on a high-fat diet tend to lose weight in studies, even if they're trying to calorie control, which shows the great benefit of high-fat diets for weight loss, but it makes the exercise data a little harder to analyse as you compare things to body weight. And then when we're talking about exercising, we can be talking about endurance tests, time trials, maximal power output that someone can attain, and their maximal VO2. So essentially, the data is quite messy. So then we came along and thought we would make this data messier by doing a completely different study as well. So our study, we have participants come in one day to the lab, and they'll do some anthropometry measures. So we look at height, weight, body composition, waist and hip circumference, which is an indicator of general health. And then we take some bloods, both venous and finger prick bloods. So HbA1c is an indicator for diabetes, looking at your glucose regulation. resting blood glucose, and then resting ketones and lactate. And then from the venous blood, we're also measuring the lipids, resting insulin and DNA. So we're looking at the CD36 gene, which is related to fat transporters in and out of the cell, which has the potential to influence how much you're able to burn. And then we also collected a urine sample, which will hopefully be used to detect fructose and sucrose in the urine, which will hopefully validate the dietary questionnaires. They then do a few different methods of nutrition analysis. Nutrition analysis is quite difficult because people tend to underreport, or depending on what they want to eat, they might change what they're eating coming up to a particular study. Some participants do admit that. I ate really healthy this week because I knew I was coming in. And then we're testing blood pressure, resting heart rate, seeing how much physical activity they're doing a week, and then they do the exercise test, which is very similar to the picture Kieron showed. So they're on a cycle ergometer, they're hooked up with a huge headpiece and a mouthpiece so we can measure their oxygen and carbon dioxide that they breathe out. And from that, we're able to tell how much fat they're burning and how much carbohydrate they're burning. So our inclusion and exclusion criteria was somewhat similar to what we used for the studies in our systematic review. There are age limits, that's just related to risk factors, and then they had to be disease-free and preferably a steady diet and weight for around 6 months. We're doing a cross-sectional study because it means we were able to get a 6-month intervention, whereas when you're doing a randomized control trial or an intervention study, just because they're so expensive, they usually only last 4 weeks, and we want to see if we have effects which are greater than which occur after 4 weeks. So currently we've tested 43 people, our aim's 100. In that mix we have 20 untrained or generally fit people, and we have 23 very highly trained people, 29 males and 14 females. And not from our own dietary analysis, but from what they identified as their diet type. We have 4 high-fat, low-carbers, 3 vegans, and 4 vegetarian pescatarians. It is great to get people who are similar because it makes it easy to compare, but in the same way, we don't necessarily have a good spread of all these across different groups. So all our high-fat, low-carbers were incredibly well-trained. Everyone who was well trained ate quite a healthy diet. Whether or not they ate carbs, they still had very, very little sugar. And then when you have the unfit population, they're the ones who are eating the bad diet. So it makes it really hard to detect the differences between the diet influence on it and the general fitness influence on it. So Kieron showed you graphs. So these are from one of the participants which came in. So on the left we have fuel usage in grams, depending on the intensity, and then on the right we have the contribution. So this is not scientifically analysed, this is just a bit of data, so it's not necessarily statistically significant, but there was a group of the well-trained group, because they're quite similar in fitness, it makes it a little easier to compare them. So this was a 10 milliliter per kilogram per minute range, and you can start to see a relationship. So the high-fat, low-carbers have been burning more fat oxidation relative to what their fitness status is. On the right in the table, you can still see there is some variation. It's quite a large age gap, but with the exception of one female, it's about a 15-year age gap. We've got 9 males and 2 females in this graph. The 2 females are the lowest data points. That's the other problem. All our fit people, minus those 2 females, are males. And then training, they're either triathletes, cyclists, or runners. We were hoping to get a lot of data for unfit populations, but these studies tend to draw people who are fitter into them. So you can see they're getting a VO2 max of, this is between 47 and 57, which is very high. A lot of people will get in the 30s. If you do maybe a little bit of exercise in a week, you're looking at the 30s. If you train regularly, you might hit the low 40s. We also had some people here who hit over 60, which is not common. They're in the top 99th percentile. So similar to the other studies, we are getting a few too many well-trained people, as much as they're really good. And then this is a comparison of their fat oxidation curve, so looking over the time period of their test at different intensities. So the previous one was just looking at their fat max. So as you can see, the graphs are a little wobbly, but there is a bit of a difference. The reason we have vegans in there is that vegans eat a very high carbohydrate diet, and some of them have almost no fat in their diet because there's so few foods they can eat to get fat sources. So that's really looking at the spectrum. And then this average is probably more towards a high-fat, low-carb diet than most of the average population. Thank you.